Welcome to this explainer. Today, we're going on a bit of a quest to physically map our most precious cargo, our memories, our thoughts, and our actions. We're going to completely demystify exactly how we investigate complex cognitive and motor processes right inside the living human brain. And we'll do this by comparing two incredible, cutting-edge neuroimaging modalities. It's a deeply fascinating look into the mechanics of the mind, and I am so excited to guide you through it. So let's just dive right in by asking a truly profound question. How can we actually visualize invisible internal processes? I mean, really think about it. How do we capture the physical shadow of a memory retrieval, or an emotional response, or a motor function happening in real time beneath the skull? For centuries, the brain was basically this impenetrable black box. But now we are literally watching it think. Here's our roadmap for today. We'll start by decoding the human brain. Then we'll look at fMRI for mapping deep structures, explore shedding near-infrared light with fNIRS, put them head-to-head in a showdown, and finally wrap up with the power of synergy. Section 1. Decoding the human brain, the orchestration of cognitive, motor, and memory processes. We really have to start by just acknowledging the brain's staggering baseline complexity. It orchestrates a massive myriad of functions simultaneously. We're talking about everything from gross motor skills like going for a run to fine motor skills like just turning a page, all the way to intricate cognitive empathy and deep memory retrieval. It is an absolute symphony of electrical and chemical activity that literally never stops. And observing these neural mechanisms isn't just some academic exercise. It's absolutely crucial for clinical progress. When we decode brain activity, we're actually advancing our treatments for severe conditions like traumatic brain injury, or TBI. Imagine a patient who suffered a severe concussion. By physically mapping the specific areas where their neural firing has been disrupted, doctors can tailor highly specific rehabilitation therapies to essentially reroute and repair those exact pathways. Understanding the map is the critical first step to fixing the roads. Moving on to Section 2, fMRI, mapping deep structures, the heavyweight of neuroimaging. Let's explore our first major technological solution. Established back in the early 1990s by Psy.G and his colleagues, Functional Magnetic Resonance Imaging, or fMRI, totally revolutionized neuroscience. Ogawa's brilliant realization fundamentally changed how we study the brain, and it has completely dominated mapping research for over 3 decades now. Okay, so the core physics of fMRI rely on something called the BOLD signal. That stands for blood oxygen level dependent contrast. The fundamental concept here is that cerebral blood flow and neuronal activation are tightly, intimately coupled. When an area of your brain is working hard, it urgently needs energy, right? So a rush of oxygen-rich blood is sent to that precise region. What's absolutely amazing is that oxygenated and deoxygenated blood have different magnetic properties. Because an fMRI is essentially a giant, incredibly powerful magnet, it can physically detect these really subtle magnetic differences between the oxygen-rich arterial blood and the oxygen-poor venous blood. We are quite literally using magnetism to read brain activity. Because of that magnetic tracking, fMRI gives us these stunning millimeter-level spatial maps of the brain. It is completely unparalleled for looking into deep subcortical structures, like the hippocampus. Remember that physical shadow of a memory we talked about earlier? Well, the hippocampus is exactly where we look to find it. But this method does have a catch. Because it relies on the vascular system to rush blood to the active neurons, there's a temporal lag. The hemodynamic response, that rush of blood, actually peaks about 4 to 6 seconds after the neural firing already happened. Plus, getting these breathtaking images requires the subject to lie completely, perfectly still inside a massive, highly expensive, and completely immobile magnetic tube. Which brings us to Section 3, fNIRS, shedding near-infrared light, the agile challenger. This is a modern alternative designed specifically to address some of fMRI's biggest limitations. Functional Near-Infrared Spectroscopy, usually just called fNIRS. Get this, the whole foundation of fNIRS relies on a genuinely wild fun fact. Human skin and bone are surprisingly transparent to near-infrared light. It sounds completely like science fiction, I know, but it's an incredible biological quirk. This means we can shine red and near-infrared light directly through the scalp and the thick bone of the skull to safely reach the underlying cortical tissue of the brain. It's totally harmless, and it's entirely revolutionary. So how does this actually work? Let's break it down step by step.
fNIRS emits near-infrared light, typically between 650 and 1,000 nanometers, right onto the scalp. The light travels into the tissue, and it's absorbed at completely different rates by oxygenated versus deoxygenated hemoglobin. Think of it kind of like shining a really bright flashlight through the palm of your hand and seeing that warm red glow on the other side, but highly advanced and mathematically precise. The machine then detects the backscattered light that bounces out. Using a principle called the modified Beer-Lambert law, researchers can track exactly how much light was absorbed, which indirectly measures the functional brain activity based on that blood flow.
Now, to get those super precise measurements, scientists use a really clever trick involving something called the isosbestic point. Let's take a second with this. This is a specific wavelength, right around 810 nanometers, where both oxygenated and deoxygenated hemoglobin absorb light identically. Why does that matter? Because scientists absolutely need a stable baseline. If they don't have a specific wavelength where both types of blood look exactly the same, they can't accurately calculate the differences when brain activity suddenly changes. By utilizing wavelengths specifically above and below this crucial point, an fNIRS system uses that baseline to independently and accurately calculate the exact concentrations of both types of blood in real time.
All right, Section 4, fMRI versus fNIRS, the showdown. A rigorous technical comparison. Now that we understand the underlying physics of both of these incredible tools, let's put them head to head. When we stack them up, spatial resolution is where fMRI absolutely wins out. It gives us a pristine, millimeter-level map of the whole brain, including those deep memory-retrieving subcortical structures. But look at temporal resolution. While fMRI is sluggish due to that 4 to 6 second blood flow lag, fNIRS operates with lightning-fast millisecond precision, literally capturing data every 0.01 seconds. However, fNIRS does have a major blind spot. It's limited strictly to superficial cortical regions. It just simply cannot see deep into the center of the brain where the near-infrared light can't reach. And these technical specs actually dictate some massive real-world environmental constraints. fMRI requires participants to remain completely, perfectly still inside a narrow, incredibly noisy tube. This often triggers claustrophobia and makes studying natural movement entirely impossible. I mean, you can't run on a treadmill or play the piano in an MRI machine. fNIRS, on the other hand, is cost-effective, wearable, and highly resistant to movement artifacts. Subjects can walk around, talk, and interact naturally while just wearing a lightweight fNIRS cap. That absolute freedom of movement is a complete game changer. Because of these widely different profiles, their research applications are completely distinct. fMRI remains the unmatched gold standard for mapping deep memory retrieval and complex resting state networks. But fNIRS? It's revolutionizing field-based research. Imagine a researcher wanting to study cognitive development. With fNIRS, they can comfortably place a cap on a toddler and just let them naturally play with toys on the floor, mapping their brain activity in real time, which is something that is totally impossible inside an fMRI tube. It's bringing neuroimaging to the bedside, to classrooms, and significantly enhancing accessibility for rural and economically disadvantaged populations who just can't access multi-million-dollar scanning facilities.
Section 5, the power of synergy, integrating fMRI and fNIRS for the future of neuroscience. Because really, if you're asking which technology is better, the future isn't about choosing one over the other at all. It's about combining their unique strengths.
So researchers are now doing something called synchronous detection. This means literally using an fNIRS cap inside an fMRI scanner at the exact same time. By doing this, scientists can cross-validate the data, directly compensating for the spatial limitations of fNIRS while simultaneously making up for that 4 to 6 second temporal lag of the fMRI. It gives us this flawlessly comprehensive picture of neural dynamics. It's just a massive breakthrough for the field of neuroscience, blending the absolute best of both worlds. And this integration, it's paving the way for one of the most exciting new frontiers in neuroscience: hyperscanning. Hyperscanning is a method for simultaneously recording the brain signals from multiple different people while they are actively interacting. Imagine wearing a portable headset while playing a board game, or having a deep, empathetic conversation with a friend, and actually watching exactly how your brainwaves sync up with theirs. It allows us to step away from isolated brains in sterile labs, and finally, truly observe the social brain in real time as we cooperate, empathize, and communicate. I'll leave you with this. As these neuroimaging technologies become incredibly portable, highly accessible, and deeply synergistic, what profound mysteries of human connection are we about to uncover next? Capturing the shadow of a memory was really just the beginning. We are now mapping the very essence of how we relate to one another. Thank you so much for joining me for this explainer, and keep questioning the incredible world inside your head.